case I don't have time, can you sit here and deal with the laptop in case you need something and somebody wants to join the meeting or something? Few more minutes, we still got Okay, so welcome everybody after lunch, and our next speaker is Laxmi Parida from Ibm Research Center. So Laxmi is very correctly on your Wikipedia page, IBM Master Inventor and the group leader in computational genomics. But that's not what Laxmi will tell us about. Today, I strongly encourage you to speak about computational genomics to Laxmi later on. But, you know, as possible career paths for computational mathematics / applied mathematicians. Yesterday we spoke to Jeff about US national labs. There are those fantastic opportunities in places like Watson Center in IBM. You can also do very nice research in this semi, I would say, industrial environment. And that's what Laxmi is. The floor is yours.
Thank you, Pavel. Thank you for the invitation, and I'm very happy to be here in Poznań. Just 24 hours ago, around this time, I was going through passport control in a very long line in Europe, in Amsterdam. And I was not sure whether I'll make it or not. I'm glad I made it. And they said, so at the passport control, they asked me, why are you going to Poland? So I said, there's a math meeting there and for a math conference. And they said, why are you having a math meeting in Poland? So I said, you know, Polish mathematicians are very strong and they are organizing a meeting and they have invited me. And then he asked me about other things and so on. But I was surprised that he was very specific. Why are you going there for a math meeting? Anyway, so anyway, I'm here. Okay, I made it, and I hear that many are having difficulties. So I feel very lucky to have made it on time. And so I want to talk to you about math, in Ibm Research. Pavel was very kind to me. He gave me carte blanche on how I will, what I will talk about. So let me begin with a message from my fellow mathematicians from Ibm Research. So this is a video that you can also find on YouTube. And I would like to say that it was made just for you. but it's actually made for you and others like you, and it has been used in math conferences also. And this particular title, I love this title. I wish I could take again credit for coming up with this title, but this is our creative directors who have labeled that YouTube as, which is in YouTube as well, which is Math is Life in at IBM Research. So it's a 5 to 6 minute video, but it gives you a sense of what the researchers are thinking about in terms of math and how they are doing it. So let's hope that it works. I mean, it seems to have my picture, but that's YouTube's doing, because if you go there, for some reason they put my face, although it doesn't begin with that. So anyway, let me play this.
With a lot of mathematics, it forces you to have a consistent formalism that brings everything together and also conceive ideas and progress the state of the art.
Math connects the intuitive ideas and concepts that we have to reality. We might have a vague idea of a high-dimensional whole in a multi-dimensional space, but it is made precise by persistent homology theory.
I'm the global leader for quantum finance and optimization at IBM Research. Working on quantum computing as a mathematician is very exciting at the moment because you can contribute to the full stack, analyzing and developing new algorithms, actually optimizing these algorithms to map them on real hardware, but also to actually operate a quantum computer.
My name is Ibm Research. I'm senior manager at the mathematics of AI. Mathematics is really the steam engine of AI. Concepts that come from statistical mechanics or even physics-oriented approach. All of these are bringing fresh, new understanding into how AI can actually work.
I'm Laxmi Parida. I lead computational genomics at IBM Research. We are in the throes of the genomic revolution. And just like in physics, math is playing a pivotal role. What can be more exciting than understanding the genome of an organism using math?
I'm Chit Apte, and I'm currently chair of the IBM Research Mathematical Sciences Council. What's exciting about math today in IBM Research is kind of its centrality to so many different threads that are of high priority to IBM in the directions it's taking. The mathematical sciences community at IBM Research is about 200 strong. We are embedded in our various technical priorities. So there are math scientists working in quantum computing, in artificial intelligence, in security. We want the math scientists to be close to the problem that they're tackling. At the same time, they work across these different priorities as a group of mathematicians who can then exchange ideas.
IBM Research is a great place to work and to do math. We can work on fundamental research on one hand side, but we also have the contact to client and business units, which give us feedback from different perspectives.
You have the flexibility to conceive new research directions and pursue them. People are excited about how much mathematics can contribute to some of those fundamental challenges that we face.
IBM Research produces immense opportunities for synergies across groups, across disciplines. There is this clash of abstract ideas with the real world challenges, which is very possible here.
Quantum computing is a fundamentally new way of computing. It leverages the principles of quantum mechanics to process information. We're working on quantum algorithms that can achieve a quadratic speedup for Monte Carlo simulation. When working on quantum algorithms, you need math everywhere to formulate the actual problems, to design the new algorithms. And often these algorithms are lightweight quantum classical algorithms where you need to understand both worlds, the quantum and the classical. We're looking at some financial applications for quantum computing, such as portfolio optimization, risk analysis, or option pricing. At the moment, we have small systems available to experiment with, but in the near future, when these systems grow, we hope to be able to demonstrate an actual quantum advantage.
Scientific discovery, we typically have some observations, and we're trying to come up with a mathematical model that would fit those points that we have. E equals mc² is something that would work here in space. Unfortunately, when you look into what the state of the art in AI, they are not generalizing very well. So they are really learning specific instances that require massive amount of data to learn. In our belief, bringing some of those fundamental deep theoretical notions from mathematics is something that can be a game changer. We are now looking into taking symbolic regression, but doing that from a principled mathematical perspective that allow us to find globally optimal mathematical formulas, demonstrating that a certain expression is indeed derivable from first principles.
Computational genomics is a broad discipline that aims at understanding biology at a molecular level based on genomics with algorithmic and mathematical approaches. So it's at the crossroads of biology, algorithmics, and mathematics. Every step of almost everything that we do, mathematics plays a big role. There are a few areas that we are working on. And in all of these, we use orthodox as well as unexpected and unorthodox approaches. We have used tensor learning in blood cancer, and we have used topological data analysis, or TDA, in microbiomes, and also melding of logic and TDA in Alzheimer's.
It's a great time to be at IBM right now because we are at the forefront of many fundamental breakthroughs that we will be making in the information technology business. Every breakthrough we are making are being enabled by advances we make in mathematical sciences. Nothing could be better than working to drive these advances that we are seeing happening today in IBM.
So I hope everyone is convinced that IBM Research, like other research labs, also is using that. Do I need to hold this? So I think this was a good opportunity for you to have. I'll get out of this one. I don't want to see my face on it, but unfortunately YouTube puts my face on this video. Anyway, it was a good chance for you to see my other math colleagues who work in quantum and who are working in AI and so on. So I as Pavel also mentioned, have been working in computational genomics for life sciences. I also chair the Exploratory Life Sciences Council. You met my math chair counterpart, which was Chidapte, who spoke. He chairs the Exploratory Mathematical Council. And so in the next few slides, few minutes, I will give you a sampling of the kind of work that we are doing at the interface of so I want to talk about how we use topology in the area of disease genomics. I'm going to call it disease genomics. And I just have a sampling of it. I'm not going to get into, my plan is not to get into the technical details. And I'm sure topology you probably understand way better than I can explain. And I will just show you how we have been connecting these things. So the first one is, I don't want to get into the details, but I'll tell you what this is. So this is on, this work is on Parkinson's disease. Okay, and Parkinson's disease, the data that we used is from Michael J. Fox Foundation for Parkinson's Research, who have a good database. And here the question we are answering is as follows: is you have the patients, okay, you have about 400 subjects, okay, you have healthy subjects and the disease subjects, and the description of the subjects is in terms of transcriptomic data. In other words, you have the gene expression profile of the patients, say of the blood. And so, and the question you want to ask is that is the expression profile telling you something about the disease? And AI, you can apply AI on it and see whether you can actually predict the state of disease or not. And here AI does a reasonable job. What this piece of work, what we did is that instead of giving the gene expression values more or less as input to your prediction system, audio AI system, we use topology on it and extract some features, topological features, and then feed that to AI, and then use that to AI. And then we find that these topological features that we are using, there is details of that, and it appears in the paper. I don't know whether you can read that or not, but if you go to our webpage, you can actually get to that paper. The details. So I'm not talking about the details. I'll just say that we use structures from topology. We use TBA, use the structures, feed it to the learning thing, and we find that it does better than without using these features. So leading us to conclude that maybe when we look at topological characteristics, perhaps we are getting something more about it. Of course, it's the next step to go further in and try to see what we are getting. What is it that we're learning about the disease? So this is an example of augmenting AI with topology to make it do better. So initially when we started, let's say, does it even do anything? And then we said, does it do as good as AI? But it actually did better in this data set and the way we applied. So that's one application that we have used. Another one is on cancer evolution. So let me tell you what the problem here is. We have a large project where we are trying to understand resistance to cancer, the drug resistance to cancer. What it means is that patients respond to a drug, and then after a while they stop responding. And we need to understand that why did they stop responding to that specific drug? And there are various cancers that one talks about. So this one specific problem we are looking at in this case that I'm showing you here is that certain cancers, the cancer evolves into another recalcitrant form, another cancer. So there is a transition that goes from cancer 1 to cancer 2, and cancer 2 is a severe cancer that they have. And of course, the researchers want to understand, clinicians, researchers together, where did the transition happen? Can we catch them at the transition time? And do we know what is bringing the transition about? Okay, so it's not very clear. Supposing you had a patient, say at month 1, he has some form of cancer, is responding to something, and when you go to say month 7 or 8, suddenly the cancer is different. They even look different under a microscope. The cells look different. So the transition has happened, and we need to understand what happened in between. So there is data that you collect. This is on single cell data. That data that you collect, longitudinal data that you collect during the transition, maybe not every day, but in a shorter time interval than before this cancer has evolved into a different form. And you look at the single cell data. So you look at all the omics data there and try to catch a transition of these cells into this other cell form. So here again we are using something. This is close to what is the mapper-like thing that is being used here. So we take this, we take the pre and the post and try to get a simpler description, because the data, as you may understand, is an extremely high dimension, and you have all the single cell data. Single cell data as opposed to bulk data is bulk data when you take, which is cheaper, is you take all the cells, squish them together and get some average expression value and so on. In single cell, you are looking at the expression value of everything of each and every single cell. And we think that maybe we'll have more information on that, but you will have a larger number of cells, and it has its own issues, is that it might have dropouts in the sense you may not get the complete profile, but you get something for finding gaps and so on and so forth. It has its own limitations, but we want to understand this transition phase where we used mapper techniques to actually catch the transition going from one to the other, and then try to see what are the features that is happening during the transitioning of this cell. We use it in melanoma. We are currently using it with our clinicians on some unpublished work right now, yet unpublished, but it's in the pipeline to get published. But mapper was used to study this transition. The next one I want to talk about, again, I thought I'll put this in here, which is again using this mapper idea, and I'm calling it a 360 visualization dashboard. So let me try to explain what the problem is, or what we want to do is that supposing you're a clinician or a researcher and you have some number of patients, 30, 100, or that number of patients, and what you're looking at, say, the microbiome data, say, or something else which is in higher dimensions. In this particular case, it's microbiome data, and you have a suspicion that this is related to the disease state of your subject. Okay, it's quite possible that you could have some data that may have nothing to do with the phenotype. So here it is the microbiome and the phenotype data. So there are two cases I'm showing you. The right top is actually with animals, with dogs, and the bottom right is the kwashiorkor disease, like due to malnutrition and so on. And there is the strong belief that there's a relationship between your microbiome and the phenotype. And now, if you have the challenge that I want to look at all the patients together, and I want to visualize all their microbiome, the essential parts of it, and their disease state or not, and all the other meta information that comes with it, their age, their gender, and everything that matters to me, their diet, and so on. I want a single dashboard. Can I look at it? Okay, and so here is a single dashboard. The top and bottom are two dashboards. Okay, it doesn't matter which one we talk about, so let me explain what they are. Of course, first we establish that there is a relationship between the information that you're looking at, omics data, which in this case happens to be microbiome data, and the phenotype in this case is disease or non-disease. Okay, so you have to establish that, and you need that function F. And then we use a mapper in this case. So what mapper has done is that it takes all your patients. So you have healthy ones here at the bottom. The green looking circles are healthy ones here, and then the disease ones, which is red. And there is a spectrum that is even in between, right? Even a clinician will say, I think this one is healthy, but it's kind of perhaps is going to develop this and so on. So that is indeed a spectrum. So your function F that you use here, that is actually associating your microbiome with the phenotype, is that function F. Then use that F for a mapper like display. So what the mapper has done, how to interpret the results here, whatever mapper does is that it has subdivided your patient space, your 100 or 200 or what number of patients you have, into overlapping sets. Okay, so you start with the healthy ones. In this in the plot, the healthy ones are at the bottom, and the disease ones are at the top. So these are the groupings of patients, and they are overlapping. The overlap is captured by the edges that you have between these disks, and then you summarize all the information that your researcher cares about there. For instance, you have and we use that in concentric circles. So the very center one says how many are diseased, healthy, and so on, based on the case. Because for the kwashiorkor, you have only healthy and diseased. For the second one, you have healthy and diseased, acute, and IBD. So there are three distinctions. And then you have a ring around it which captures the statistics of your patient in terms of gender, age, what diet they were having, or anything else that you want. So it is giving you for that little group. And of course, you have the healthy, non-healthy, and so on. So we use this for a non-trivial visualization of a group of patients, which we think captures all the information that a researcher or clinician looks at. And how they use it is probably up to them, but this actually puts all of the patients together, very high dimension data, and so on. Here we use microbiome. It's possible to use other kind of dimension data. Yeah, so the next one I have is on, right, this is in metabolic syndrome. This is actually not on genomics, it's on phenomics. What it means is that for every subject, you may have their molecular information in terms of the genomic data, but you can also have a lot of information on the phenomic side or the clinical side, what the clinician, the data that the clinician is gathering. It could be BMI, it could be your blood pressure, it could be a various other bunch of information that is collected. And then you want to look at this and try to say something about the disease etiology of your subject. So this one is a work that actually takes this your phenotype descriptions and says how it contributes to the pathogenesis. It is able to pull out some what I call composite phenotype markers, some disease markers for this disease based only on the phenotypes. Yeah, so there were some details of this, which I'm just going to skip. Yeah, so these were the use cases. There is also a Matilda, a system, a toolbox that we are working on. Some of my team members who are working on it, maybe you saw them in the video that they were in the discussion, in the video discussion that we are doing. So this is a multi-purpose toolkit for TDA. So this is something that we use in all the applications that I was showing. And if we needed some special structures for what we need, maybe tomorrow, if some of you are there, I'll talk a little bit more about what are the other structures that we need. So this toolbox helps us with our specific needs, and of course, we intend to make it available when it is ready for the community as well. And before I sort of wrap up, I wanted to also mention other ongoing work. The left-hand side is on a combination of AI and topology. Okay, so this is some attention networks, higher order attention networks, which is using topological ideas to enhance graph neural nets. Okay? And on the right-hand side is combining quantum and topology. Okay, so this is on quantum topological data analysis with linear depth and exponential speed. So there are many ways you can combine. These are archived, so they're accessible. This is not one of them is from my group, the other is not, but you saw some of the authors in the little video that I showed you. So hopefully this gives you a sense of maybe the breadth of the work that we are doing. The first four, the disease and omics, was my specific group and what we are doing, and this is the bigger group that is how they're bringing in AI and quantum and other things with topology. So the next the next part of the session, I wanted to keep it as a Q&A or a discussion with you if you had questions or if you have comments and other things. So I wanted to keep it interactive. And
can we go back to the example with Parkinson?
Oh, Parkinson's, all the way to Parkinson's. Yes.
Would you explain more details? What did you do there?
So, okay, actually, I should have had a few more slides on this one. I just have only one slide, so let me tell you roughly what we do. So this is, you have a training set and you have a test set, right? So for the training set, what we did is we went and looked at all the profile of the expression for the training set, and then we kind of built a gene skeleton, okay, a skeleton of those, and then we take the values of the expression values into account and do a filtration by sort of growing these balls in those little spaces, and that's how we bring in the trained data onto. We are using filtration on the skeleton that was kind of built on the on the trained data, the test data expressions we bring in to get the filtrations on this skeleton, and that filtration is used for the neural nets, convolutional neural net. Yeah. I know I was not very clear on the details, but it gives you a sense of how it was used. I think we can look at I will point you to the papers and you can see, or we can discuss later about it. Yes, this is a published paper.
Paper, but the presentation.
Oh, the presentation. Yeah, I can make it available. Yeah.
The talk tomorrow. Sorry? You mentioned the talk tomorrow. Yes, that's right.
Yeah, but I may not be covering this. I don't think I'm covering this, but it can be other stuff there.
Yeah, so that's not the purpose, but so when you're speaking about the skeleton, you're doing like this compressed RIPS complex of some sort?
Yeah, so the Viator is RIPS complex. Yeah. I should have gotten some few more slides. I unfortunately don't have them, but I thought this is going to be a very general level.
I take it before because I asked you to speak about in general They work in a research institution, but not an academic research institution, but some commercial, not about details of the research. So that's my take. There was a question over there.
Okay, thank you very much for that. Are there any internships available within your team?
I'm sorry, so could you say that again?
Are there any internships available, like short-term work with your team?
Internship?
Yeah.
Yes. And summer interns, for the summer, we usually take very large number of interns. In fact, currently, as I speak, we have a few hundred interns in the research lab that you saw in the first video. And the internship is usually during summer, but there are probably there are exceptions which I have to kind of find out. Yes, we are open to taking interns. Yes.
So do you also take interns with your team, the TDA?
Yes. In fact, I currently have 3 interns. One of them is working on topology, and the others are working on the genomics. So my team does take, and the other team leaders that you saw about using the right. So they also do take interns, and the total number of course varies from year to year, but we very strongly hire interns. In fact, I myself, I just complete the story. So we have different programs that are called interns, externs. There's also something called co-ops, and I'm not sure whether we currently have them or not. It is while you're doing your PhD, you spend time at Ibm Research. And I did my PhD at NYU, New York University, and I went and spent a year in IBM while I was a student, and that was a different program. I have to check that kind of program we still have or not. And I did that, and after that, I joined IBM. So I've gone through a similar route. Yes.
I think you already just answered half of my question. So I wanted to ask, what career levels are the internships aimed at? So PhD students, postdocs?
Okay. The internships are for PhD students or master students. We also take undergrads. Okay? So the internship is for students, undergrads, graduate students, either masters or PhD students. For postdocs, it is a postdoc position. For postdocs, it will be a postdoc position or what is a research scientist position, based on people's experience and so on. So that is a different route. While interns we hire in extremely large numbers, during the summer and other times maybe. The other one is a little more restricted. Depends on what are the openings and what exists.
Sorry, can you please explain a bit more the advantage of PDA methods compared to the traditional, I don't know if it is traditional machine learning methods to tackle your questions?
Right. That's a good question, because that's a question we ask ourselves always. I personally like sort of PDA and kind of try to apply it. And the first, the way we have been working is we try to see where can we map TDA. It's not very easy to map our problems into TDA. Let me say that out front. So when it comes to topology or TDA, because it's a data analysis there, topology, most of the problems that I'm talking about from the genomic space, they're kind of in an abstract space, right? I'm not talking about the 3D proteins that we'll take a protein structure and say, can I find a hole in that or not? To me, I think that mapping is a little simpler. Okay? Most of the data that I talk about is in a sense abstract. So it's in a high dimension, it's in some abstract space, right? Because when I have 20,000 genomes, my data is lying in a 20,000 dimensional space. And then I'm trying to see what I'll get at. And it is not always obvious how you're going to map your problem to the topological tools that are there. And we want to take the existing tools. And sometimes the tools don't do what they want to do, and we want to go and change it around, right? So, but we want to take these tools and apply. So that's the first hurdle. So we have a deep interest in this, and we take it. So tomorrow we're planning to attend at Bedlover. Bedleo, sorry, I'm sorry. At Bedleo, I plan to give those applications where I think that it outperforms classical methods. So for us, it is an exercise. When we say what is the advantage, it is not always clear. And also at the stage that we are in, it's not that topology says that we can compute more efficiently yet, right? And that's why in IBM, we are trying to say, we are actively looking at quantum computing, and whether quantum computing is going to help us in, handling scale in topology. You probably understand better that scaling is a problem even in topology right now. So it's an active research area for us to handle this, and then say topology did something that classical methods did better. And I have not yet come across an application where I always compare it with classical methods, and I show that it is done better. In all our papers, that is our general theme, because we don't want to say why we used it. It should do better. But I haven't come across a problem where only topology has actually given you an answer where a classical method, when I say classical kind of AI, it doesn't answer. In the Parkinson's that I was showing you, I said that using topology actually gave us better results, did better prediction than, the slide is still up. Not in all cases, not in all measures, but in most of the measures, it did better. But as you can see, it did minimally better, right? It's not out of the ballpark or something like that, but it did better. So it is an exercise and a struggle and a journey and a path. Let's put it that way. Thank you.
Yeah, I mean, if I don't drop in, so even in machine learning, there are no proofs to show that on average neural networks are doing better than random forest. You see in practice in many instances, it's the same with AI. If you add it, oftentimes you get something better. Okay, so I want to steer a discussion because Laxmi still wants to play one video. And I think what we have over here is we have a crowd of very bright master PhD or very postdoc level people, including some of the older, let's say, or more senior. Poznań Research Center, it really looks like this. I mean, so the place is really amazing, and that's a nice opportunity to work. Actually, can you tell us more about this internship options for various levels? Maybe what do you expect from people when you are interviewing? What sorts of skills you are looking for? If one of our guests over here would like to comment.
Right, that's a good question. I will tell you from my point of view how it has worked sort of with me. And usually there is a call for interns, and IBM has a relationship with the universities, and they announce that, and it shows up in the university boards and so on where the students apply. And if people know me or people who are hiring, I get a little bucket. You saw that there are many researchers from this huge pool. Everybody is given a little bucket, right? And from that bucket, I get 1 or 2 or 3, depending on that year and how many interns we are taking. I also reach out to my contacts and say, do they recommend their students, or is there someone? So usually the process has been, the interview process for internship, the way I've seen it, has been informal. It is through, I will trust your professor. Sometimes I've already worked with students, and I know how good they are. And then I work with the professors who give me their CVs. I put it into a common pool, and I kind of advocate for the candidates that I want. And this is the process. And sometimes I do, of course, I do talk to students if I'm not completely familiar with. So the interview is a kind of, we do have to fill in all these things. So we do talk to them. It's an informal process. It's more on, I know that if students want to learn and other things, and the professors think the students are good. So we trust that, and we have a short interview, which is not very intimidating. And then we pull it out from the bucket, and then one is picked. So the usual thing is that when I have a bucket here, I have 3 openings. I can take 3 candidates. I might have 4 or 5, say, or 6 or 7 or 10 CVs. I will take the ones that align best with my research, and then I'll pass it on to the other researchers also in case it works. So it's an informal process. Get in there early and apply. And it is international. We are based, by the way, I should have probably clarified. We are based in New York, in the US. Yeah. So yes.
It's about 45 minutes
from the city, but it is. We have a lot of interns who stay in the neighborhood. Some live in the city, even employees. Some live in New York City and can drive up. But the location is in New York. I know we do take international interns as well. And the other thing I should clarify: we have labs all over the world too. So the research lab that I use, that I am from, and that you saw here is in Yorktown, near New York City. It's kind of the headquarters of research lab, but we have labs in Dublin, in Zurich, in Haifa, and in many other countries. About 11 or 12 labs.
Actually, when you're looking at the CVs, what are the skills you are looking for?
Okay.
That's not top secret.
So I have some, when I am looking at, for example, when I have looking at the CV, I'm taking them for some purpose. You know, I have a problem that I want to work on. Supposing it's a topology problem. In fact, this year I did take a student and he's working on topology. Then if I'm looking at the topology thing, I will look at the topological aspect. We also want them to be able to code, do programming, and so on. So that is also, these days, is becoming a skill that one must have. So some of your coding skills and some of the background, specifically what I have, usually the researcher has something in mind is going to assign. It is not that the interns pick a problem, okay? It is usually not the case, unless the intern has a very exciting problem and mentions it to your mentor or the hiring manager, and this is okay, let's look at the problem. Usually we already have some specific problems in mind, and we're looking for the interns to move in. So the CV, what we look at the CV is the usual stuff, your programming skill, and the other areas where which area you're going to work in at the lab.
Okay, so you mentioned a collection of big programs that your group is working on. But if you have a particular intern coming to you, what this year will be working on?
Right. No. So when I say big problems, some problems are broken up into sub problems, right?
Okay.
Okay. Oh, by the way, the Parkinson's work that you have been asking was the work with a summer intern. Summer intern and some of my team and me and so on. But the work was kind of driven by a summer intern. But the problem we gave it, so we have this Parkinson's data. We want to use this. We were already looking at it. So that was the work with a summer intern. Now I'm trying to think what were the other pieces I presented. No, that was the work with the summer intern. Tomorrow I'll present some other works, which some summer interns take the initiative and drive the work. In fact, tomorrow I'll talk about a work where she learned the basic things that we are doing, and she drove the work. I said I'm interested in the COVID-19 data in this TDA, and she completely drove the work to publications, papers, and so on. And I'll talk about one of those tomorrow. So when I say drive the work, there's a little bit of connection there, in the sense that this is the data that we are grappling with. We want to understand this data, and these are the different tools we are looking at. Can I use TDA? Can I use AI? Can I use bioinformatics? Can I use algorithms? We are kind of playing with it, and the intern can come in. He can drive the work up to the intern, and and we always have mentors and so on. So we are working together.
So many problems we expect, other skills we are expected to have, but mostly in this room we are non-US citizens. Is IBM helping with getting visa and all this bureaucracy stuff for the non-US overseas citizens?
Yes, because our program is international. I've had interns from Italy before. Okay, and I'm trying to think of other European countries. And because I knew faculty there, and so that was the connection. So we have had interns from abroad, and that was a while ago. And I know from out of the 200 interns that we have, many are coming from abroad. Of course, it will depend on the situation, and of course, IBM will completely. If we go through it with it, and if I get the green light, IBM will do everything in time, so this has to start early on to get your visas and everything in place. IBM is very good. I guess so are most of your hosting institutes with that. So once I get the green light that this is fine, there is something that they look over to. One is a technical aspect. The technical aspect, it's up to me, right? I decide that this student is a good fit for the work we are doing, is going to be valuable both for the student and for us. That's the technical part. And then the other non-technical part is this kind of visa things. Will they get a visa? Will this work or not? And what are the hurdles? We go through that process. Once we get a green light, I think we'll do everything for you to get there.
Maybe a private question. So if a student would like to ask you a short question, like a paragraph, can I fit in your group, or maybe one group of the colleagues would be okay if they send me this short email?
Yes, they could, but you see, this summer it is already a little late, right? It will be for the next summer, because this summer we are almost at the mid time of internships are all happening at full swing. I said 200 interns. In fact, due to COVID-19, researchers were not coming into the buildings. We're working remotely, and for last 2 years we had remote internship because of the pandemic. And this is the first year we have in-person interns. So the interns have landed, and now they outnumber the scientists, because the scientists are still trying to work safely from home or something. But they are there, they're in the building. And if you have an intern, and I have mentors, we do physically go into the building and work with them. So yes, for next year, certainly, and it will also depend on when we call out for the interns, but it's good for me and others also to have your CVs already in place so that we can move fast when we are told when we have your needs.
So Laxmi mentioned that she also still wants to show a 15-minute video, so I think it's a good time to do it.
Sure, right.
Unless there is any quick question, but it has to be quick.
Yes.
Just the video is the first one.
Just could you, in 30 seconds, say what's the biggest difference to work in a private funded research center with respect to, either in academia or, like we saw yesterday, a national lab? And what's the relationship between the corporate side of IBM in this case?
Right. So I don't know what you were told from the national lab side. I'm sure you were told great things. I'm going to say we are great, equally great or even greater. So from the corporate side, that's a very good question. So the corporate we do research to support the corporate. Our funding is coming from the corporate side, right? So we are kind of dependent on that. But we have enough freedom to do our research in different areas. So the big level areas will be recognized, like quantum, math, and that kind of subject areas, and now future of computing and so on. So that's why I wanted to show you the next slide, which is coming from our leadership, as to IBM is always looking forward, like most other places. You're looking forward. What's going to happen in the next 10 years? Okay, what is happening now? We are under control and we are leading it. But what's going to happen in the next 10 years? We are looking ahead. And what are the technologies that will lead us there? So in AI, it's quantum and so on. So the next slide, the next video, which is for 15 minutes, is coming from sort of our leadership, where they are sharing their dreams and the paths that we are going on. And we are kind of aware of that's the bigger direction. So as long as we are sort of in that bigger direction, moving in that direction, we have enough freedom to do research in that area. Does that answer your question?
There's not much difference from the grant. There are hot topics in which are getting the grants right now. There are others which we are not. So it's really not much. They don't have to be quick.
It's very good. When is the best time to apply for next year?
Okay, so it depends on the cycles there. It is say around January, February is when we begin to get to know for this summer, which will be April to May. So around January, February time, we start sending out our feelers or start seeing who are the candidates. Approximately that time, like I said, it will differ from year to year.
Internships start when?
Internships usually start in the summer at mutual convenience around May or so. And we always make exceptions if there are little things, but say around May to August.
Yeah, there's a problem because the academic year in the US and EU is a bit shifted.
Yes, so we can always shift those around. Okay, internship is generally standard time is for 3 months. Okay. Usually we try not to do less than that because it's too small to do anything. But there are extensions. Some people try to do longer. So 3 months is the kind of time, and reasonably well paid also. I don't know the details, but well paid to survive in a high cost of living there, but well paid to be able to handle it. And your travel and everything is covered. The visa, travel, and everything.
Okay, sorry, I need to cut it short. So there will be time during coffee break.
So this video I'm going to show you is to answer almost your question also, is the bigger dream or the bigger vision that we have had across in technology and where we are moving.
I've always been a little bit obsessed with understanding how the natural world around me works. My parents instilled in me the importance of questioning everything. My father used to always ask me about things that he would observe in daily life. Can you figure out how this works and explain it to me? And I think like that inquisitive nature might be why I chose science. It was a way of satisfying that curiosity to discover.
We find ourselves in a moment of enormous potential in our technology, but also extraordinary challenges that it would be hard to overstate how deep and profound they are.
On one hand, if we just think about the challenges that the world is facing in sustainable development goals like water, poverty, food, healthcare, and climate.
Climate change is also my upbringing. Now, I came from a developing nation where I have seen the impacts of climate change,
and we were all witnessing the impact of a global pandemic and the realization that it will probably not be the last one.
We didn't have the luxury of decades to solve that. We needed to find a solution fast.
In healthcare, it takes 17 years on average between the time when scientifically we know that something can work to the time when it's available for use.
And we are just building knowledge so quickly, it's become very difficult, almost impossible for even scientists to keep up.
If we can compress the times to discovery, we can find solutions to some of the most pressing challenges that we're facing today.
Those are daunting, massive challenges, and without accelerating the rate at which we discover solutions, there is no path to be able to address these fundamental challenges.
We really don't have much time to waste.
Ibm Research is a community of scientists, engineers, and designers of all sorts of disciplines. We have mathematicians, and physicists, and chemists, and experts in AI and quantum. And we come together to create and imagine new ways to process information and generate the next advances in computing. Ibm Research has played through our history and continues to play a central role of creating new solutions and imagining new ways to bring computing to solve some of our hardest problems. And if we weren't here, I think we would lose a massive vector of progress for the world. I'm Dario Hill. I'm Senior Vice President at IBM, and I'm responsible for all of the research activities of IBM worldwide.
I'm Saya Nazario. I joined IBM Research because I wanted to make quantum computing a reality. We need to put together the power of all these new technologies that are emerging.
The moment we find ourselves is the convergence of not just one technology, but 3 technologies that are going to alter how we practice science and how we develop technology. I like to summarize them as bits plus neurons plus qubits. The world of bits is the digital world, the computers that we all know. enter the world of artificial intelligence. That's the world of neural networks that have inspiration from the biological brain, and they allow us to solve problems differently, to learn from data. And then enter the third element of the equation, the blend of physics and information, the world of qubits.
Which is the promise of quantum computing.
All combined together.
With that purpose and focus to discover faster.
Our objective is to accelerate the rate of discovery by a factor of 10x. So we want something that would have taken 10 years to do in 1 year, or something that cost 100 billion to do it in 10 million. I think what the world hasn't grasped yet are the implications of the convergence of these technologies in the context of accelerating scientific discovery. We really have set ourselves a target of not a little bit better, but really a significant jump.
We have hybrid cloud. We have quantum computing that's on the horizon. There's a lot of progress that has happened in AI. I think it's going to be important for us to figure out how we're going to make them more accessible so that scientists do what they do best, which is really focusing on tough problems. My name is Salman Asafa, and I'm a vice president at Ibm Research. I lead strategy related to climate and impact science. Ultimately, the vision that we have is how do we develop the tools, the framework, and the hybrid cloud-based platform so that it's accessible for someone with just a computer and an idea. Let's say we have a scientist in the lab, a university, fresh postdoc. All she has is computer, some simulation software, and some ideas. She could use deep search to go through years of publication. She doesn't have to download all of the documents. She can just access the cloud. Instead of a few months, it could be a few hours. That's where the power of the cloud is.
AI is a very important and powerful capability that allows us to harness data in new ways to achieve tasks that were previously impossible. So one of the key things that our teams at IBM are working on are AI technologies that can ingest scientific articles. And essentially the computer, in some sense, is reading those articles. And that trains AI models to help advance the scientific process. The role of the computer here is really to augment human capability.